is the river of life, carrying the secrets of health and diseases. Welcome to AHS Guide, your one-stop destination for clear, concise, and exam-focused lectures for the 5 subjects in first year of Allied Health Sciences. Here, we break down the complex concepts into simple and easy explanations with notes and diagrams, and as well as quick revision tips, helping you to learn smarter and not harder. So in today's session, we'll be studying from the pathology. We are stepping into the fascinating branch of hematology, which is the study of blood, blood-forming organs, and the related diseases to the hematology. We'll explore how the blood cells are produced, their structure and functions, and what happens when the disease disrupts the delicate balance of this normal body blood cells. From anemia to leukemia, clotting disorders to infection of the blood, the hematology not only reveals the story of our health but also plays a key role in diagnosis and treatment. By the end of this lecture, you'll be able to understand why the blood tests are required to uncover what type of disease that is causing behind the pathology of it. So the contents of whatever we are studying today is Introduction to Hematology. We'll be seeing the normal constituents of blood, their structure and functions, and the collection of blood sample as well. The various anticoagulants that are used in hematology, the hemoglobin estimation, different methods and normal values, packed cell volume, erythrocyte sedimentation rate, normal hemostasis, and blood bleeding time, clotting time, prothrombin time, activated partial thromboplastin time. So here we will be studying the same topics as in physiology of blood. The same thing will be covered, as well as the diseases that are related to the blood types and blood cells. Now, the introduction to hematology. The scope of hematology is concerned with the diseases of the blood and bone marrow. So it also includes the study of circulating blood components and the blood forming organs. So hematology is basically the study of the various diseases that are related to the blood and bone marrow. These blood and bone marrow also consist of the blood components and the blood forming organs that are there. So we will be studying about the circulation of the blood components inside the blood forming organs and the blood vessels. The component of study of blood cells are RBCs, which are red blood cells, which can lead to normal and abnormal counts of the RBCs or the diseases in the RBCs, for example, anemia, polycythemia. White blood cells and the diseases related to it like leukemias and lymphomas. For the platelets, thrombocytopenia, thrombocytosis, bleeding disorders like hemophilia and von Willebrand diseases, and blood groups and transfusion related diseases that are essential for safe transfusion practices. So these are the components of study under the under the hematology topic. The WHO classification of World Health Organization classified hematology based on both circulating leukocytes and tissue resident leukocytes, wherein the leukocytes that are circulating in the body or the tissue resident leukocytes wherein the blood components are inside the tissues and causing the diseases. Based on the concept that the blood and tissue immune cells are interrelated, can migrate between the compartments. So these leukocytes can be migrating inside the blood or the tissue immune cells and causing the diseases. We'll be studying about them as well in hematology. Now, how hematology is studied. So hematology can be studied in 2 forms. One is laboratory and clinical hematology. Under laboratory, it involves the study of the tests, which includes the complete blood count, the blood smears, the bone marrow biopsy are used to understand hematology. Clinical hematology is where the diagnosis and clinical features and management of the hematological diseases involved. So laboratory wise, you check the blood count of the patient or the blood smear or the bone marrow biopsy of the patient. Clinical hematology involves the using the tests, what are the diagnosis that is made, how the clinical features are like the signs and symptoms, and the management of hematological diseases with the treatment or the various other ways to manage the diseases. Now, the goal of hematology to be studied is under basic principles and management of the strategies for common blood disorders. So, what are the basic principles of the common blood disorders and the management strategies that should be adopted is the goal of the study of hematology. to gain the knowledge in diagnostic techniques and interpreting in hematology. So in order to understand the disease, the knowledge about them is essential in order to diagnose and interpret the type of disease that is seen under blood cells, abnormal blood cells. The normal constituents of blood and their structure and function. Hematopoiesis is the production of formed elements of the blood. So hematopoiesis is the process of formation of blood cells from the bone marrow. So these blood cells differentiate and become pluripotent, thereby producing different types of cells. So where does these blood cells or hematopoiesis takes place is inside the bone marrow. Bone marrow is present in between in the longest bones of your body. So the bone marrow is present as a tissue that helps in pluripotency of the different cells of blood. Circulating blood normally contains three types of mature blood cells, which are red blood cells, white blood cells, and platelets. Red blood cells are also called as erythrocytes, white blood cells as leukocytes, and platelets as thrombocytes. These blood cells perform various physiological functions. Erythrocytes are involved in the oxygen transportation. Leukocytes are involved in the body defense mechanism or the immunity. They act against the infection and tissue injury. Whereas thrombocytes are involved mainly in the blood clot formation and maintaining the normal blood flow inside the blood vessels. So thereby preventing the blood loss as well. So the three components are red blood cells, white blood cells, and platelets, which are erythrocytes, leukocytes, and thrombocytes. The functions or the physiological functions of the respective cells are erythrocytes or RBCs are involved in oxygen transportation and carbon dioxide bringing back to the lungs, and leukocytes are involved in body defense, which is immune system, and thrombocytes are involved in the blood integrity, which is inside the blood vessels, as well as while the injury has happened, preventing the blood loss is also involved, is the function of a thrombocyte. Now here is the image of how a pluripotent stem cell, which is present inside the bone marrow, divides into two types. One is myeloid and the other one is lymphoid. The myeloid is a trilineage stem cell because it can further divide into three different types of cells. It is called trilineage stem cells, and lymphoid stem cells divides into one type of cell. So we'll first see the myeloid stem cells. The myeloid stem cell can be further divided into granulocyte, monocyte progenitor, erythroid progenitor, and megakaryocyte progenitor. Granulocyte monocyte progenitor gives rise to the different WBCs that are present inside the body. Erythroid progenitor gives rise to the red blood cells, and megakaryocyte progenitor gives rise to platelets. There are various intermediates in here. Under granulocyte monocyte progenitors, we have three types of granulocytes and one type of agranulocyte, which is monocyte. So granulocytes are neutrophil, basophil, and eosinophil, and the agranulocytes are monocytes. Now under erythroid progenitor, we have, in the presence of erythropoietin, RBCs are present. RBC cells are produced, and the megakaryocyte progenitor, with the presence of thrombopoietin, can produce platelets. So these are the myeloid stem cells generation, and lymphoid stem cells produces pro-lymphocyte, which further gives three different types of cells, which is B lymphocytes, T lymphocytes, and natural killer lymphocytes, which are all produced by various regions. B lymphocytes and natural killer lymphocytes or NK lymphocytes are produced in the bone marrow, whereas T lymphocytes are produced in the thymus. So this is a division of how a normal blood cell that is produced produced in a normal human adult. It is important to understand the generation or the pluripotency of the stem cell, which is helpful to understand the diseases that are causing at each stage of development of a different blood cell. Now RBC, the first component of the blood, the major contribution of red color to the blood as well. So RBC structure and characteristics. So RBC, to see it is biconcave disc. You can see, observe the image, it is biconcave shaped, and the size is around 7.2 micrometer and thickness 2.4 micrometer, and at the center it is 1 micrometer. The nucleus and organelles, it is absent in mature cells. So in the mature cell, nucleus and organelles are absent because they make space for oxygen to be carried inside the cell, RBC. The main components are of the RBC are 90% or more than 90% there is hemoglobin by weight, and the flexibility, it can squeeze through the thin capillaries as small as 3.5 micrometers, and the lifespan or the length amount of days it can stay alive is 90 to 120 days, and it is commonly accepted as approximately 120 plus or minus 30 days. Now the functions of RBC is, the first function is oxygen transport. The primary function is to carry oxygen from lungs to the tissue. And how does it do? By the oxygen binds to the hemoglobin A and a tetrameric protein, which is a tetrameric protein, and binds with the oxygen and is cooperative in affinity, causes the sigmoid O2 dissociation curve, allowing the oxygen to be transported from the lungs to the tissues. The value of the partial pressure of oxygen is said to be around 50% in the HB, which is the saturation of the oxygen tension at HB is 50%, and hence it is called P50. The higher the P50, the lower affinity and easier the oxygen release. So the 50% of hemoglobin oxygen tension should be at that range, and anything higher than P50 is said to be lower affinity, allowing the oxygen to easily break down with the hemoglobin and flow through the tissues and allow the tissues to absorb the oxygen from the RBCs. Now the influence of oxygen release is how the oxygen is released from the hemoglobin attachment is by globin chains, pH, and sometimes by 2,3 bisphosphate glycerol. Now globin chains have hemoglobin A and hemoglobin F. So hemoglobin F has lower affinity with oxygen compared to hemoglobin A. So HB in adult releases more O2 to the tissues, allowing easier dissociation of oxygen with the RBC and leading to oxygen transfer from RBC to the cells. Now pH is happening by the Bohr's effect. If the blood is said to be having lower pH or acidic, if the blood is acidic in nature, then lower oxygen and hemoglobin attachment, thereby releasing more oxygen to the tissues. So if the HB and O2 binding is lower, it is easier for the oxygen to be released from the RBC to the tissues. So pH plays a vital role, and acidic pH helps in easier release of oxygen to the tissues. 2,3 BPG, which is bisphosphate glycerol, wherein increased BPG, as in case of anemia and hypoxia, the HbO2 affinity is low and the enhanced oxygen delivery is also seen. The carbon dioxide transport is another function of RBC, wherein the tissues releases carbon dioxide. The tissues released carbon dioxide is accepted by the RBCs and sent back to lungs. How does the mechanism happen? Here CO2 binds with the hemoglobin of hydrogen bicarbonate present in the RBCs and diffuse into the plasma. So inside the HB does not absorb the carbon dioxide, but the bicarbonate ion absorbs the carbon dioxide and allows the diffusion into the plasma. The pulmonary capillaries or the lung capillaries will convert this bicarbonate ion into carbon dioxide and is removed outside in the form of exhaling the carbon dioxide. The carb amino HB, sometimes the CO2 binds to the deoxy HB, that is the HB which is not combined with oxygen and is a reversible compound when released and is released in CO2 in lungs. So the thing that you have to understand in CO2 transport is that there are two methods of release of CO2 in the blood. One part is that it can be converted into HCO3, that is bicarbonate inside the RBC, and transported is diffused into the plasma and later transported to the lung capillaries, wherein the CO2 is exhaled outside. The other type is the carbaminohemoglobin, which is the Hb, or the hemoglobin present in the red blood cells, combines with CO2, which is a reversible compound, is released to the lungs as a CO2. One is carbamino-HB and the other one is bicarbonate. There are two products that are formed after carbon dioxide is released from the tissues to the lungs. The energy metabolism is the third function of a red blood cell. The anaerobic glycolysis, or Embden-Meyerhof pathway. Here the ATP is generated for the cell survival and produces the 2,3-bisphosphate glycerin, and NADH or NADPH via HMP path shunt. The red cell function and maintaining redox balance is important. So how does the energy provided? How is the energy provided to the red blood cells? Is by anaerobic glycolysis, which is the EMF pathway. Here the ATP is generated for the red blood cell to survive through the process. Since the red blood cell does not have its own oxygen, the EMP pathway is followed over here, and the 2,3 BPH allows the ATP generation from NADH and NADPH, which is important for the red cell to function and maintain its redox balance, reducing and oxidation process that is happening while accepting of oxygen and removal of oxygen. The NADH and NADPH is formed. This NADH and NADPH provides 2 to 3 ATPs at one cycle. Now the lifespan during the circulation of the red blood cells have various lifespans in circulation. While circulating, the neutrophils can live up to 8 to 12 hours inside the blood vessels, and for platelets about 8 to 10 days, whereas RBCs can stay up 90 to 120 days while circulation. Now homeostasis is the process wherein the blood cells production is tightly regulated to match the cell loss and maintaining a stable count unless altered by the diseases. So homeostasis is the process where the blood keeps the cell count at a constant rate and is not allowed to either decrease or increase the cell loss. So to maintain it, the stable counts are kept by the body. If it is altered, there is chances of any other diseases inside the human body. That is what homeostasis means. WBCs, or white blood cells, are further classified based upon the presence of granules in them. Granules are the small tiny structures that are present inside the cell's body. The main types based upon the granules are two types: granulocytes and non-granulocytes, or non-granule leukocytes. Then in granulocytes, they contain cytoplasmic granules, that is the granules that are present in the cytoplasm of a cell. In non-granular leukocytes or agranulocytes, there are no visible granules under the microscope. Now granulocytes can be further divided based upon the nuclear appearance, that is how the nucleus is. They can be classified as polymorphonuclear leukocytes and monocytes. Granulocytes are further divided into two types, which is polymorphonuclear leukocytes and the monocytes. The polymorphonucleocytes, or PMNs, are further divided into neutrophils, eosinophils and basophils. The neutrophils are having granule colors which are pale pink or lilac, and the role of neutrophils is the first line defense and phagocytic in nature, that is they consume the whole foreign object or microorganism that is causing disease or infection inside the human body. So hence they are called the first line defense in the human body. The second type of PMNs are eosinophils. They are bright red or orange color granules present inside the nuclei, and they usually play the key role in allergies and parasitic infection, thereby causing the reaction against allergies inside the body. Either fever or redness, rash, itching is happening because of the release of eosinophils inside the body. The third type of PMNs are basophils, which are deep blue or purple in color. They release histamine, which are against the allergies and cause the inflammatory reaction. Now the Other type of granulocyte, which is monocytes, these have largest WBCs and they have kidney shaped nucleus. They are larger than the WBCs and are kidney shaped nuclei. The rest of neutrophils, eosinophils and basophils have multi-lobed nuclei. That is, there are many lobes of nuclei present in them. The monocytes are the precursors of macrophages and dendritic cells. So they are, monocytes can further be divided and the cell forms a macrophage or a dendritic cell. The macrophages are the ones that engulf the foreign objects or the microorganisms that causes diseases, or dead cell tissues are eaten up by the macrophages. Dendritic cells are the ones present in the nervous system or the neuronal system. Now the monocytes are involved in phagocytosis, that is what the engulfing of the foreign object or the cell debris, and antigen presentation, that is the presence of antigen on the body, which are involved in antigen antibody reaction, causing the immune system to react. Now non-granular leukocyte, the second type of WBCs, which is agranulocytes. These WBCs can be further divided, or lymphocytes can be further divided into T cells, B cells and NK cells. T cells are cell-mediated immunity and also such as cytotoxic T cells. These are involved in cell-mediated immunity. Their T cells trigger the monocytes to be released. In turn, monocytes are going to be released as macrophages or dendritic cells and involved in the immunity of the body. B cells are involved in production of antibodies against the antigen consisting microorganisms. These are humoral immunity. NK cells or natural killer cells are involved in destroying the virus infection or tumor cells, which help in innate immunity. All these cell-mediated immunity, humoral immunity and innate immunity are explained in the physiology class of blood chapter. You can go to the video and learn about them in detail. Now white blood cells, as I told, the pictorial representation is here. The monocytes, which have single large nuclei and they are involved in the antigen and phagocytic nature. Eosinophils have pink granules in them, pink or lilac granules, and they have a headphone shaped nuclei or bilobed nuclei. So there are two lobes of nuclei. Here basophils, they are multi-lobed and they have various lobes inside the nucleus. They have blue colored or purple colored granules in them. The neutrophils here have red or orange granules in them and they are also again multi-lobed nuclei. Here are the lymphocytes, B and T lymphocytes with large nuclei inside them. So those are the different types of WBCs. Now polymorphs, which are a type of WBCs. The first type, which is neutrophils, we'll see about the general features of neutrophils. They are 12 to 15 micrometer in diameter. The nucleus is dense and has 2 to 5 multi-lobed nucleus. Now the cytoplasm has pale with fine violet or pink granules in them. These granules are going to determine what work they are and how they appear. So there are different types of granules in neutrophils again. The primary granules are called azurophilic. Their appearance is large and coarse, that is rough in appearance. Their appearance is seen in promyelocyte stage, and the contents of the granules are, these granules contain hydrolases, elastase, myeloperoxidase, capsaicin G and cationic protein. They also have permeability increasing protein and defensins, which are micropicidal proteins. You just need to know about the general features and the types of the granules, that's it. Not extra contents are not required for the time being. Now the secondary granules, we saw the primary granules of neutrophils. We have the secondary type of granules, which are specific granules. Their appearance is small and many in number. The stage of appearance is again myelocytic stage, and these are MPO negative. The contents of these granules consist of lactoferrin, NADPH oxidase, histaminase, vitamin B12 binding protein, receptors for chemoattractants, and laminin. So you can have the basic understanding that the contents of the granules act as the defense system against the foreign substances or bacteria that is causing infection. The function of neutrophils is chemotaxis, phagocytosis, and microbial killing. So chemotaxis is against the chemical signals that are causing the infection or inflammation inside the body. So the movement towards these neutrophils moves towards the infection causing site and are usually attracted by the chemical signals that are produced by the microorganism. Phagocytosis is engulfing and digestion of foreign particles or microorganism. It acts as like macrophages, not macrophages. So macrophages, as in case of monocytes, in case of neutrophils it is smaller compared to the macrophages. Microbial killing, again the oxygen dependent mechanism is involved here. It involves reactive oxygen species and myeloperoxidases and cause the oxygen independent mechanism. Thereby the enzymes act as defense either by lysozymes, which cause lysis of the cells, or proteases by degrading the protein present inside the microorganisms. These are the various ways the organism is killed. Now the lymphocyte, the type of a WBC. Under lymphocytes we have peripheral blood, which consists of various features. The size of lymphocyte, smaller lymphocytes is 9 to 12 micrometer, larger ones are 12 to 16 micrometers. The nucleus are slightly round or indented. In large lymphocytes it is the same type. The nucleus are round and slightly indented. The chromatin is coarsely clumped. Again, large lymphocytes also show the same type of signs. And cytoplasm is scanty and basophilic. Now the types of lymphocyte, as I told before, there are T, B and NK lymphocytes. Plasma cells, they originate from the B lymphocytes and upon stimulation by the B lymphocytes alone. The nucleus of the plasma cells are eccentric, that is eccentric in position, meaning they are in the center. The cartwheel pattern of chromatin is observed, the chromatin which is involved in cell division. The cytoplasm consists of deeply basophilic and pale perinuclear zones, that is the Golgi apparatus. Plasma cells under normal presence, that is normal peripheral blood smear, it is difficult to see because of the absence of any granules in them. Whereas in myelomatosis type of disease, you can see the many number of plasma cells present in the blood smear. Now reactive lymphocytes is another type of a lymphocyte wherein it is also known as Sturck cells or plasmacytoid lymphocytes. They appear in viral infection, that is the plasma consisting of reactive lymphocytes is only seen in viral infected patients, and they are usually basophilic cytoplasm and are similar to plasma cells. The type of lymphocytes and their functions, we'll see the different types of lymphocytes in the body. T lymphocytes, which is thymus dependent or it is originating from the thymus. So the maturation happens in the thymus itself and hence it is called thymocytes or T lymphocytes. The immunity type is cell mediated. Again, cell mediated immunity is dealt in physiology chapter of blood. You can go and see the cell mediated immunity in detail over there. The surface markers, again CD3 plus, is present in T lymphocytes. The subtypes of T lymphocyte is cytotoxic T cells and helper T cells. Cytotoxic T cells are CD8 plus and kill infected or abnormal cells, whereas helper T cells are involved in coordinated immune response, that is they cause delayed hypersensitivity. CD4 plus is going to cause increased number of release of various other cells like monocytes and macrophages. That is why it is called helper T cells. Cytotoxic T cells directly involved into the infected or abnormal cells and kill them. Now B lymphocytes are bone marrow dependent, or maturation takes place inside the bone marrow itself. And these B lymphocytes can be differentiated into plasma cells. That is, B lymphocytes can mature to form a plasma cell. The function of B lymphocyte is to produce antibodies or immunoglobulins. The IgG, IgM, IgA, IgG, all these different antibodies are released and controlled by the B lymphocytes. The immune type is humoral immunity. That is, the humoral immunity is dealt in the physiology chapter of blood again. You can go back and review the humoral immunity there. The surface markers of B lymphocytes, that is the component that is present on the cell membrane, is varying, and it might usually have CD19 plus and CD20 plus proteins. So these are the proteins that are present on the cell surface. Now, NK cells, the last type of lymphocyte, which is natural killer cells. These are innate immune cells and appear like lymphocytes. These do not have cell markers like T lymphocytes and B lymphocytes have CD8, CD4, CD19 plus and CD20 plus. So there is no cell marker like them. And the function of natural killer cells is to directly kill the virus infected in tumor cells, and they are usually part of natural and innate immunity. So it is innate immunity is taken from the mothers or the cells directly from the environment. Now the monocytes, the other type of blood cell, which is agranulocytic in nature. Here the size is largest mature leukocyte is around 12 to 20 micrometers, and the nucleus is usually large, oval, indented, sometimes notched or horseshoe shaped. They have fine reticulated chromatin and is less clumped than the lymphocytes. Here the cytoplasm is abundant, the cytoplasm is heavily put, and the cytoplasm is pale blue in color due to the presence of dust-like granules and vacuoles inside the cell. The function of monocyte, or the role of monocytes inside the body, is to phagocyte, again engulf pathogens and debris, antigen presenting cells. Thereby these monocytes trigger the release of T lymphocytes and act against the antigen presence of the cell. They are involved in adaptive immune responses, and these are also mediators of inflammation, thereby allowing the release of cytokines and prostaglandins, which are involved in the inflammatory response. These also stimulate liver to produce some acute phase proteins to deal with the inflammation. Now, the precursors to tissue macrophages, they also act as precursors to tissue macrophages. That is, the monocytes also trigger the release of macrophages, which are involved in digesting or deleting the cells that are causing damage to the or infection to the body. So macrophages can be seen as as various types of cells in different organs, whereas in liver it acts as scuffer cells feeding on the dead or infected liver cells. Microglia are the macrophages present in the brain, eating on the dead tissue or the infected tissue of the brain. Alveolar macrophages are present in the lungs, allowing the digestion of the foreign objects or microorganisms that is causing infection to the lungs. From the above part, you can understand the reticuloendothelial system or mononuclear phagocytic system. So these are the different RES systems that we are going to be learning from monocytes. RES is reticuloendothelial system. The red blood cell as well as monocyte that are involved in phagocytic nature of the blood system. So as I told, the monocyte has a big nuclei which can be indented, as in here it is indented. The cytoplasm is abundant, more number, and also has pale blue colored cytoplasm. There is also lysosomes present inside. These lysosomes involve in digestion or attack of the foreign objects. Now, eosinophils, a type of granulocyte, which is generally 12 to 15 micrometer in size. The nucleus is typically bilobed, and the cytoplasm is coarse or deep red granules are present because they are acidic in nature. They stain intensive with peroxidases because of the high content of oxygen present, and they can stain only in the presence of peroxidase enzymes. Now, the granules have the components of basic proteins like peroxidases, which cause high staining. They have cell adhesion molecules, cytokines, charcot-laden crystal proteins. So each component of the granule have certain function or significance. Like basic proteins are involved in being toxic to the parasites and pathogens. Peroxidases are involved in oxidative reactions. Cell adhesion molecules help in eosinophils migrating to inflammation sites. The cytokines promote the eosinophil growth and activation. The charcot-laden crystal protein is seen in asthma, forms crystals in the lung tissue and deals with them. So each granule content or component is involved in various other functions. Now the common function of eosinophils is immune response to the parasite, like the worms of helminthes, and the granules are cytotoxic to these parasites, thereby killing the parasites inside the human body. They also modulate or regulate the allergic reactions which are involved in hypersensitivity and asthma. They counteract against the effects of histamine release by the basophils and mast cells. So the histamine that is released from basophils and mast cells react with the asthma and cause the allergic reactions. Either the excessive sneezing, fever, or redness that is caused as a reaction is because of these eosinophils. Now, antigen-antibody reaction is also regulated and controlled by the eosinophils. They participate in immune complex processing and tissue repair. These eosinophils are increased in number when there is a parasitic infection or allergic condition or eosinophilic disorders as well. This is an image of a eosinophil. Now we have basophil. Sorry, this is an image of a basophil. We have already seen the eosinophils. No, eosinophils is this. Now basophils have general features such as mature granulocytes. So these are said to be mature granulocytes because of the irregular or bilobed nucleus and obscured by granules, because the basic nature or the maturation of granules present here. Here the basophil is 12 to 15 micrometers and the cytoplasm is filled with coarse intensely basophilic or granules, and they are above the nucleus and make it hard to see the nucleus. The function of basophil is to mediate allergic and hypersensitive reaction, and it majorly involves in type 1 hypersensitivity. For example, asthma and anaphylaxis are the reactions acting reactions against the immune infections that has been caused. These basophils involve in release of histamine and degranulation and cause the classic allergy symptoms like rash, redness, or fever, etc. The inflammatory response is happening by allowing the immune cells to be present at the site of injury or allergens. The parasitic defense is less significant and assist in the eosinophils dealing with the parasites. So the basophils versus mast cells, here the features are the location wherein the circulation is in the blood, whereas mast cells reside inside the tissues. The origin of basophils is in bone marrow derived. Mast cells also are derived from the bone marrow. The granule consist of histamine, heparin, and 5-HT in basophils, whereas mast cells, they have the same type, which is histamine, heparin, and 5-HT. And the functions of basophils is allergic reactions, and mast cells, it is allergic and local inflammatory response. So this is an image of a basophil, how it looks like. You can see that there are blue granules present inside the cell, and there is a bilobed nuclei, and there are the blue granules are so heavily present inside the nuclei that they sometimes cause difficult to see the nuclei under microscope. Now the platelets, the last type of blood cell after RBC, red blood cells, and WBCs, the platelets. So in these platelets, the basic function of these platelets is, basic features of these platelets is they have 1 to 4 micrometer in size. They are discoid shaped and non-nucleated like RBCs as well. And granules, they have red or purple stained granules under microscope, that you can see red and purple stained granules. The normal count is 1,50,000 to 4,00,000 per microliter. The range is 1,50,000 to 4,00,000 per microliter. The lifespan is around 7 to 10 days, and its distribution is 70% in circulation and 30% sequestered in the spleen. That is 70% platelets are seen in the circulation of the blood and 30% are dead in the spleen. So platelets can be in two forms. One is discoid shape, the normal round shape that you see here. These are in normal state, but when they are activated or when they are starting to work, they are like this. They form hair-like structures and they are activated for clotting. Now, platelet functions are mainly to maintain the hemostasis. Now, hemostasis can be divided into two main phases. One is primary and the other one is secondary. The primary hemostasis is platelet plug formation, and the secondary hemostasis involves the coagulation cascade. Now, here the primary hemostasis is where the temporary plug is formed when a bleeding is happening at an injury. Now, the time frame of primary hemostasis is immediate. It can happen within seconds to minutes. So the goal is to produce a rapid stop of bleeding from the small vessels. The steps involved in the primary hemostasis is platelet adhesion, platelet release reaction, and the platelet aggregation. So under platelet adhesion, the platelet adhere to the collagen of the damaged skin tissue, which is endothelium, and they cause the GP1A and GP2A receptors to release the von Willebrand factors. These von Willebrand factors attach to the GP1B and 9th complex of the factors that are produced in the body. So these are clotting factors that are present inside your body. 1A and 2A and von Willebrand factors are the present inside the body. Whenever an injury happens, the collagen that is exposed has near the damaged endothelium allows the platelet to adhere to the collagen and produce all the factors like von Willebrand. So when von Willebrand is released, the more amount of platelet release is activated. So the second step of primary hemostasis is platelet release reaction, wherein the platelet is activated and more number or dense granules are required and released into the body. And when the more number of platelets are released, there are various other mechanisms also happening, like dense granules is released like ADP, ATP, calcium, serotonin, alpha granules like PF4, factors like 5 and 8, von Willebrand factors, PDGF, fibrinogen, thrombospondin, and lysosome enzymes are also released in order to facilitate the hemostasis or the platelet release reaction. After the platelets are released in more number, the platelet aggregation happens, wherein the fibrinogen forms the bridges between platelet and with the GP2A and 3A receptors, they form a temporary clot or a temporary plug at the site of bleeding or injury. This is the image of how the primary hemostasis happens. Like I told, there are endothelial injury, then the exposure with the platelet adhesion. Exposure is where platelets go and see the collagen, and collagen along with when platelets bind to the collagen, there is release of von Willebrand factors and platelet adhesion takes place, wherein the platelet activation also is caused due to release of many number of platelets and the presence of von Willebrand factors. Once the platelets are released in more numbers, a thread-like structure of fibrinogen is formed in between the platelets, forming a temporary platelet plug. So the main goal of primary hemostasis is to prevent or reduce the blood loss that is happening. Now secondary hemostasis is second step, wherein fibrinogen is produced. Fibrin is produced by the fibrinogen and perform or reinforce a protein mesh in spite of the temporary plug that is formed over here. Here the protein mesh is formed and a hard clot is formed over there, thereby forming full recovery of the healing of the injury. Now secondary hemostasis, as I told, it's a coagulation cascade wherein it might take several minutes to cure the clot, temporary plug. Now here the goal of secondary hemostasis is to stabilize or to keep the platelet plug that has formed temporarily into a permanent clot form by forming a fibrin clot. Here it involves the plasma coagulation factors to form a fibrin mesh that will form a permanent clot, fibrin clot. Now the collection of blood samples and various other topics that are involved later are dealt in another video. So we will be completing the hematology with the normal structure, function, and the clinical relevance of various blood cell components. The rest of the topic will be dealt in another video. Till then, subscribe to the channel and share it among your friends. Let them also know AHS guide is going to help the allied health science students to understand the concepts easier with the topics that are given by Rajiv Gandhi website. Thank you.